Thank you very much. Thank you very much for your presence here today. So first, I must apologize because at the time I wrote the abstract, I didn't know that I had only 15 minutes. So I wanted to present two examples of hard work, but I would have to present only one. I'm sorry about that. So I'm not a bioinformatician, so I'm a little bit outsider here. I'm more a systems biologist, but I think it's very nice because we could collaborate in a lot of different projects and stuff. So my name is Rodrigo Mora, and I want first starting perhaps introducing this field of systems biology, explain a little bit what it is. Sometimes it's hard to really grasp what is this concept about. So at the beginning 2000s, we had a lot of data already, genomics, proteomics, metabolomics, and people started to consider this should be all integrated in one concept called systems biology. So this proved to be mistaken, actually, and later it had actually adapted some other definitions. Now it's considered like a pyramid like this, where we have bioinformatics as a source of input and data for computational biology, and computational biology that can be extended towards systems biology. So here are some definitions. Systems biology is not the biology of systems. That's hard to grasp also. It is those new properties that arise when you go from the molecule to the system. It's different from physiology or holism, which studies the entire system. It's different from introductionistic things like molecular biology, which only studies the molecules. It is the in-between. Right, this is the working flow in systems biology. Basically, we start with a hypothesis, and we can even draw our hypothesis on a piece of paper, thinking how our system is behaving. What is our system about? From here, we have to create a model, so then we have to give some mathematical expressions for this. And then we start a process of fitting or parameter estimation, where we have to evaluate here the goodness of fit of our models. So basically, most of the times, our models are wrong, so we have to refine here and start iteration until we have a good model able to explain our data. Once this is happening, so this is great, we can continue, we can move forward, and then we can say, okay, we can use this for analysis and model prediction. We can design the experiments, generating experimental data, and we can then input this back here in this perhaps broader cycle. Many times we need a combination of approaches. So this is what we call a bottom-up systems biology approach, when we start from network models, or a top-down systems biology approach, when we start more from high throughput data. And many times we have to meet that in between to study these emerging properties. I will start talking about microRNAs. Why microRNAs? So you know very well, this is the Nobel Prize of Medicine this year. And this is done by Victor Ambros and Gary Ruvkun, who were the discoverers of microRNAs. And this is very interesting. It's a very nice example of scientific collaboration, because one of them discovered a target, another one had the microRNA, and they talked to each other, and then they realized they were working with the same system indeed. And now, so this has exploded in the fields, and now there are thousands of microRNAs discovered. But what are those microRNAs? They are like small molecules. They could be 21 to 23 nucleotides, or base pairs. They are synthesized in the nucleus, of course, and they have all these different mechanisms, and I'm not going into details here, where a strand of the microRNA will go and alter gene expression. So there are basically two mechanisms. One is acting in a translational repression way, and another one is degrading messenger RNAs. But there are also some other related mechanisms that have been more recently discovered, and they're also related to this. And why is this promising? This is promising because there is a whole field for therapeutics also. And so there are many diseases for which there are some microRNAs that are very signature microRNAs that could be implemented as a targets for therapy. And there are also clinical trials where the microRNAs could be regulated. So down-regulated using anti-miRs, or it could be up-regulated also using microRNA mimics. So it's nice that you can regulate this in both directions and not only with drugs that you can block things. But usually microRNAs are considered sole regulators of noise in gene expression. So they could adhere at different levels. Here we have an example without microRNA regulation and with microRNA regulation. And that is why many people, especially in these last years before the Nobel Prize, started to neglect microRNAs because they were considered only as regulators of the noise. But the question came then, what happened if they played together with transcription factors? Because you know transcription factors, as you've seen in the previous talk, they are also regulating gene expression. And these are just some examples of what happens when these two types of molecules play together. So we could have negative feedback loops that would render would lead actually to robustness, the property of robustness in the cells. We could have positive feedback loops that will lead to bistable switches. We could have microRNA clusters with coordinated responses. And those that are actually exclusive for this type of networks, coherent feed-forward loops, where we could have signal prolongation, incoherent feed-forward loops, where we could have analog digital converters, and we could have target hubs for signal integration. So you can imagine now here how the cell is adapting all these regulatory circuits or regulatory motifs to respond to the environment. The problem is this. So this is more or less how the networks look like, and of course, that is why we need a computer. The human genome contains almost 2,700 mature microRNAs. 60% of the genome is regulated by microRNAs, estimated to be regulated by microRNAs. Each microRNA can influence hundreds of targets, and each target can be targeted by several microRNAs. So this leads to very, very complex networks. It's time for a picture. There, okay, there you go. Perfect. You give me these 30 seconds at the end. So, and that's why we had to develop our own biocomputational platform. This is called BioNet UCR, which is open for all of you to use. And what we do here is we start with a list of genes of interest, and we have compiled and updated also some databases of microRNA transcription factor interactions also with their targets. And what this what this platform is doing is automatically generating these network topologies that you can open using Cytoscape, for instance. But also, it transforms this into a SBML model. This is systems biology microbe language. This is the microbe language that we use in systems biology. And also, we have there are sources of data, such as the NCI60 data, the Cancer Cell Encyclopedia, and the TCGA in cancer. So for this, then we can import this SBML model into Copasi, which is a top tool in systems biology for parameter estimation and many other tasks that I will show you later. So these are two examples I want to talk about today. So this is basically how biocomputational approaches to generate these networks and these mathematical models. One of the examples was this macrophage multi-stability, and the other one was the work that we have done on gene dosage compensation. So we just give one slide of the first example that I cannot show you today, but we have reviewed this in this review that we have here, this multi-stability macrophage activation pathways and metabolic implications. So you can have a look there if you're interested in this. The role of microRNAs in these regulatory networks and the potential bistability in macrophages. And now we're talking about gene dosage compensation. This is the hurricane that we have now affecting some of our countries, and probably will give me like a couple of days longer here in Medellin, so I'm very happy about this. But this is not the tornado I wanted to show. I wanted to show this here. So actually, this is a comparison, an analogy we're doing between genomic instability in cancer and a tornado. So we have an increasing, like this current of air that's going upwards that could represent genomic instability. So you see like increase in the number of chromosomes and the dosage of chromosomes, and therefore gene dosage. But there is a core that it's represented here by this current of air that is going downwards. And we hypothesize there are some mechanisms that could keep the genes in the level, so like a certain level that is not toxic to the cells. Because sometimes, especially for genes like MYC, you can have 7 copies of the gene, but actually the expression levels are not really increasing that much. They're only increasing a little bit, and this has some pro-oncogenic function. But if this would be unleashed, and then we have like a really overexpression of the gene, the cell could die. We have reviewed this also in this in cancer medicine, in case you have interest in this topic. So basically, I'm going to present the results of this work that we have done. This is called MYC dosage compensation mediated by microRNA transcription factor interactions in an nuclear cancer. And here we have started with a list of candidate genes. We started here with very basic criteria using coefficient of variations in between the DNA, the RNA, and the protein. And using our platform, we generated this kind of network topologies. And there we have thousands of these types of regulatory circuits. We have the oncogene MYC here. You can see how many interactions MYC is participating in. And I have to clarify, all these interactions are experimentally validated interactions from databases. So this is not predicted interactions. So we have constructed the model. We have done this parameter estimation. This is the objective function that reflects the difference between the experiment and the simulations. And then in the end, we could obtain a model like this, where we have the experimental data, the simulated data, and the difference, which is non-significant. And already when we have the model, we can do other experiments. For instance, this is a parameter scan, where we can artificially increase the MYC copy number or the copy number of the genes. And then we can evaluate whether the gene is dosage compensated, like this here, this is MYC, or it's not dosage compensated when we have this linear increase. Once we have a mathematical model, we're also able to do other types of controls. This could actually answer a little bit of the question of the previous talk also. So like, how do we know what is the most robust target within a pathway? So we can do sensitivity analysis, and then using this, we can tell which transcription factors or which microRNAs are regulating mostly one gene within our network. Then we started to do some biologically inspired experiments. Here, for instance, we could repeat the experiment, we could increase MYC copy number, and then we could check which genes are also being upregulated together with MYC copy number, or which genes are also downregulated. We had similar results for STAT3, but let's focus on MYC. So here we have another experiment where we could block specific molecules, and this was very interesting because we could block, so like if we detach MYC from the system, then we see like a linear increase of MYC. But if we block any of the other individual genes there, so we still remain blue here. This means we are still under dosage compensation. So that was a bit strange at the beginning, but then we performed other experiments that would be impossible to perform in vitro. So this is our control condition here again, in response to increasing MYC copy number, so we have dosage compensation. If we block all the transcription factors, we still have dosage compensation, but if we block microRNAs, then we lose dosage compensation. But now using this condition, now we are able to restore the function of individual microRNAs. So in this condition, we could then restore microRNA 17 and recover dosage compensation. The same with microRNA 19A and microRNA 28, and not with these others here. So then in the end, we realized that these three microRNAs were independently able to perform gene dosage compensation, MYC dosage compensation. Therefore, using the control conditions, if we block these three microRNAs, then we lose dosage compensation. So integrating these results on MYC and STAT3, we could come down to this minimum model of gene dosage compensation for MYC and STAT3, where we could have here three feedback loops with these three microRNAs, which actually belong to the same microRNA cluster, and also here some other regulatory motifs for STAT3. This small model, actually, it's able to recapitulate the behavior of this huge model that I showed you earlier. And this is interesting because actually the presence of feedback loops in our network topology is not enough to predict this property. These properties actually depend on the kinetic parameters of the motifs. For instance, MYC has 120 feedback loops, but only 3 of them are able to compensate. So this is analysis of the kinetic parameters. So this is the parameter landscape. And here in the blue region is basically where we have found motifs that are able to compensate dosage. And here in red will be then appearing all these other loops that are not compensated. So this is very nice, something here, but this is only in the model, so we had to come back to the lab. Luckily, we have the cell lines to doing these experiments, but unfortunately, there was no technique for this, so we had to develop our own technique to validate if gene dosage compensation or MYC dosage compensation was real. For this, we developed a genetic tug of war. This is like this game where there's a group of people like pulling the cord in this direction, another one like competing. And here for this, we have overexpressed an exogenous version of MYC, which is actually differentially coded because it has different like codon codification, but the same protein product. Like this, we were able to differentiate it by PCR or real-time PCR. And in the presence of the exogenous MYC, we could demonstrate the downregulation of the endogenous MYC. This is a direct demonstration that there is a regulatory circuit, and we could also see an increase of the corresponding microRNAs. So coming back to the model here, then it was interesting also to assess what is the effect of blocking MYC dosage compensation by downregulating these 3 microRNAs in the case where we have a MYC copy number of 2, 4, or 7. And as you can tell here, this is actually the control condition with 2. This is it would be make a diploid condition, so like the effect is very low. But if we have increasing copy numbers of MYC, then the effect will be much higher. And MYC, although it is an oncogene, is reported to have also toxic effect once it's overexpressed. So then we came back to the lab, and then we did exactly the same experiment, but with real cells, let's say in vitro. And then we should demonstrate that cell lines with increased MYC copy number are more sensitive to the blockade of MYC dosage compensation. And here in the end, we could also check some TCGA data from patients. This is breast cancer patients. We could separate them in 3 clusters, one of them which had no amplification of the gene, others with high compensation, and others with low compensation. We could see here was that patients with a model showing high MYC dosage compensation had actually lower survival probabilities. In these cases, here we could also demonstrate that this microRNA-19A was the principal agent, or microRNA promoting dosage compensation. So the conclusions from this first example, that will be actually the only example today, so is we have developed this platform called BioNet UCR, called to model large microRNA transcription factor networks. Cancer cells must evolve mechanisms to cope with an output, including gene dosage compensation. The compensated genes have low variation in expression despite high copy number variation. We have developed this minimum model of MYC dosage compensation for MYC in STAT3. This dosage compensation depends on kinetic parameters to emerge in silico. We have also developed a novel technique, genetic top-of-work technique, to validate transcriptional at the transcriptional level. And we believe that the blockade of gene dosage compensation may represent a novel target against amyloid cancer. So this is the graphical abstract of the paper. So this we have a microRNA 1792 in the cluster here, and we have now some other studies on this. But I wanted to finish showing this slide here, and then the acknowledgements. And this is a potential way to collaborate. And I think if you have a list of genes of interest, and if you're interested in analyzing the layer of microRNA regulation on this list, so we could actually input this and generate this kind of networks. You can also use gene co-expression analysis to enable network simplification. And then to train the models here, you can think on the usage of time and pseudo-time resolved data. That can be very nice to have these dynamic models for simulation, sensitivity analysis, and stability analysis in the cases where we are studying bistability. So we are also very interested in studying bistability, and this is the work we have done on macrophages that could not present today. But bistability is also a very broad phenomenon that can be studied in many different systems. So I want to acknowledge my international collaborators in Germany and the US. So Arregnier, Castellas, and Pedro Mendez, the support from the GEU in Mainz, our lab at the University of Costa Rica, CICA, and the funding from Alexander von Humboldt Stiftung, the NIH, CREA, and CONADE in Costa Rica. So this is our research center in Costa Rica. You are more than invited. So we have a clinic for early cancer diagnosis, a fully equipped lab, including single cell facilities, genomic facilities, sequencing, flow cytometry, cell culture, pretty much pathology, pretty much everything there. And a training program on minimally invasive surgery also, where we also have the OncoPic model. So this OncoPic model where you can grow tumors in the piglet model action. So these are my contacts here, and I think I have this. Also, of course, I want also to draw your attention to our master program on bioinformatics and systems biology at the University of Costa Rica. I actually just opened today, and they could they're accepting new students here. And this is my last slide, I promise. So this is our research center at night, and these are some nice pictures from Costa Rica, just to invite you there, and then tell you that we have 5% of world biodiversity in only 51,100 square kilometers. Thank you very much for your time. I hope you like it. Questions? Thank you.
Well, I have a couple of questions. The first one is about distribution of loci coding microRNAs in the genome. So are they, is there a pattern? Are they like in tandem, regulated by one single promoter? Are they expressed? Are they coding introns? Do you know?
Okay, so in general, long non-coding RNAs?
No,
microRNAs. Okay, yeah. So microRNAs, they are everywhere through the genome. This is interesting actually. They are a little bit more concentrated close to the fragile sites of the chromosomes. So it's not precisely known why. We're trying to study also in this direction. And that's kind of mysterious why they're mostly concentrated there. So most times they are part of genes So they could be also co-expressed with genes. Sometimes they have their own promoters, and sometimes they are, sometimes they are also in the intron. So like it's not a trend. They're everywhere, basically. Yeah.
That's one thing. And the second one,
Yes.
you were talking about transcription factors that are conserved across all animals, from corals, sponges to.
microRNAs too. microRNAs too.
And those microbes, are they?
Very conserved, very conserved. That's why it's also very interesting, this from the Nobel Prize. They are so conserved, and these regulatory pathways are very interesting. So actually, so the microRNA layer is very conserved, but if you go to the link RNA, this is not conserved. So like, for instance, if you compare like apes to humans, so microRNA is basically the same, but link RNAs are very different. So that's where like this fine regulation takes place. This is very interesting. Yeah, very nice questions. Thank you very much. Yes.
You talk about the assumption of stationary phase. What is the implications in the biological discussing
Yeah.
We assume about the stationary state.
Yeah, thank you for catching this. Actually, with the low amount of detail I gave, wouldn't realize that someone would actually catch this, but this is very nice. Very good question. So we start our approach assuming that each cell line or each case from a patient is a steady state of the system. Okay, this is just a way to have like many different samples of potential steady states of the system. But the best is, and we have done this with other data sets, that you can train your models with time resolved or pseudo time resolved data. So basically, when you're studying bistability, it's okay if you study only like some cases of one steady state here, another cases, another steady state here, but then you don't know the precise trajectory from one steady state to another until you have really time or pseudo time resolved data. So I would say if you are studying like things or like systems where you can assume steady state or quasi steady state approximations, it's fine. You just work only with this like static data. But if you have time resolved data, this is always the best in system biology, definitely. Yeah. But the question then, the answer to your question would be like, it depends on the system you're studying, on your experimental question. Yeah. Tell me.
So considering that the microRNAs can target certain genes, have you considered in your model the co-regulation, like co-dosage regulation? Like not only one gene, but more than one gene?
Yes, that's a very nice question. Thank you. So I don't have time to show this, but we have 800 candidate genes, dosage compensated, across the cancer genome. So this we're about to publish soon, hopefully. We don't know the mechanism for all of them, so we think we are only grasping really the tip of the iceberg, and so we have found like potential mechanisms for about 10 genes only. But as I told you, there are 800 genes, and some of them, so we believe like, for instance, MYC could have like a layer of regulation, like it's under the influence of MYC, and therefore we can have like also dosage compensation there with other like regulated genes directly.
Because of MYC, not necessarily because of the microRNAs. I'm talking about the microRNAs.
Yes, exactly.
Yeah.
Exactly.
So
This we can see also.
If it is because of the gene that is dosage regulated or because of the microRNA.
Yeah, but this is easy to do because you have the model there. You can then basically block the corresponding interactions and assess what is the behavior once you have fitted their model, basically. Yeah, thank you.